Excuse me. It's not sharing. It is? Yeah. Okay, so I want it on slide to show. This. Thank you. Thank you. So, if you don't remember anything, remember this cartoon. When you have a parasitic interaction, one organism is always doing the giving. The other organism is doing the taking. And the taking is such that the organism giving is in distress. That's why you have tears coming out from the rhino's eyes. So that is essentially the concept of parasitism. It's an unequal, unbalanced interaction between two different organisms, okay? So now we know what parasitism is in terms of the interaction. How can we make sense of the myriads of parasites that we can have infecting a human being? The basic definition: the parasite is the organism that derives benefit, usually to the detriment of the host, which is the organism that harbors the parasite. Now, when we say parasite, they can be located either within or on the body of the host. When they are on the body, we say they are ecto, to depict that they are outside the body system. In such a situation, we say they are causing infestation. So ectoparasites lead to infestation of the host. An endoparasite is the parasite that lives within the host's body. So they cause an infection as opposed to infestation. Is that clear? Now, in terms of how parasitic they are, we refer to some parasites as obligate, that is, they cannot exist without the host. There must always be a host for that parasite to exist. So that's an obligate parasite. A facultative parasite can switch between living on a host or living independently. So it's not obliged to always be a parasite. Do you get the point? So we can have obligate and facultative parasites. We can also have what we call accidental parasites. These occur when the parasite is not found in the usual host. Due to some form of accident, in quotes, the parasite finds itself in a different host that is not its usual host. As a result, the parasite cannot develop to its adult form or complete its life cycle, but it can still cause infection or cause trouble in the accidental host. So take, for example, you are all registered medical students in Al-Faisal. If you go to King Saud University, they don't have your records. Although you are a medical student, but you are not their medical student. So you can't sit for exams, you can't graduate from King Saud University. You have to come back to Al-Faisal, right? So the time that you spend in King Saud, you are an accidental medical student. Do you get the concept? Good. The host is the organism that is losing out in the interaction. And just like we have definitions for parasites, we can also define hosts. When we say a host is the definitive host, it means that is the host in which the parasite undergoes sexual reproduction. Or, because in some parasites, we can't really say this is the sexual reproduction stage, we look out for the adult or the most developed form of the parasite. So the host in which you find the most developed or the most matured form of the parasite is the definitive host. The host in which asexual reproduction occurs or in which the immature form of the parasite occurs is regarded as the intermediate host. Excuse me. We also have what we call the transport host. Now, unlike the host in which you can have development, you can have maturation and all, the transport host plays no role whatsoever in the replication or development of the parasite. It is just there for a mechanical transfer of the parasite from point A to point B. The parasite undergoes no development whatsoever in a transport host, okay? A reservoir host, however, keeps the parasite viable and in a form in which it can be transmitted to another susceptible host. So there's some form of biological interaction between a reservoir host and the parasite. Although the reservoir host may not reveal overt symptoms of infection, okay? Just like you can have accidental parasites, you can also have accidental hosts. So these are not the usual hosts for the parasite, but somehow the parasite got in there and is causing trouble. Another term that we use frequently in parasitology is life cycle. And what does that mean? Before I explain the meaning, let me give you the concept of the term life cycle. As human beings, our life started with fertilization of the male and the female gametes, right? Then we went through the 2-egg stage, 4-egg stage, the blastocyst, then there's implantation, then we had the embryo, then the fetus, right? Those are the stages of development we went through. Because we are higher life forms, the stages of development are a little bit complex. With parasites, these are simple organisms. So the stages of development is not as complex as that of human beings or animals. So when we say life cycle, we are referring to that sequence of development that occurs from an adult form of one parasite to the next adult stage. So when we talk of human development, it starts when an adult male and female have sex, and then there's fertilization, right? So it always starts from the adults that are sexually matured. So it's only the adult form of the parasite that can replicate. As a result, when we talk of life cycle, we always start from the adult form of the parasite. Is that clear? Then the different stages which can occur in one host or in two hosts or more than two hosts, depending on the parasite you're dealing with. But whatever the number of hosts involved, when we say life cycle, we are referring to those stages of development that we move the parasite from one adult form in mature stages, then the next adult form. Now, in terms of life cycle, you can have what we call direct and indirect. What does this mean? With indirect life cycle, as shown here in the diagram, you have at least two different organisms involved in the transmission and maturation of that parasite. So take, for example, the malaria parasite. It requires both the mosquito and the human being to complete its entire life cycle. Whereas for some other parasites, all the stages of development occurs in just one host. So that's direct. You need just one host. Indirect. In indirect, you need at least two. One of the hosts is now referred to as the intermediate host, while one is referred to as the definitive host. And remember, the intermediate host is the host in which you find the immature form of the parasite. The definitive host harbors the matured form of the parasite. Okay, so now we know a little bit about the terms and what they mean. We know why we have, this classification of parasites, commensals, mutualistic interaction, and things like that. We know what all this means. But why are you being bothered as medical students to learn about parasites? One is because parasitic infection is a global health problem. Some parasites are associated with malignancies. For example, schistosoma hematobium is associated with cancer of the bladder. Some flukes, which are parasites, flatworms, are associated with gallbladder cancer. So these are infections that could lead to very devastating health problems globally. And there are factors that can promote parasitic infections, which, if your public health system is really, really standard, should be able to prevent transmission of these parasitic agents. So factors such as poverty. With poverty, there is reduced access to healthcare. People don't present early for treatment, and so you can have very devastating consequences of the parasites. With malnutrition, you can imagine in young children, particularly children below the age of 5, and in pregnant women who are not well fed, and in addition have parasitic infections. These parasites are competing with somebody that doesn't have enough food for that little food. So it worsens the presentation. Then we have high population density. With high population density, social infrastructures may break down. Access to pipe-borne water, safe disposable or disposal of waste and things like that may become a problem. Living in crowded areas will enhance transmission of parasites from an infected person to the next uninfected and susceptible host. Now, there can be lack of personal hygiene, which is linked to lack of potable water. Imagine if people have to walk like an hour to access stream water, maybe they will only have their bath once in a week. So things like these are factors that can promote parasitic infection. And of course, when you have weak government policies, such that you have poor environmental hygiene, poor housing infrastructure, poor health facilities and things like that, all these will promote parasitic infections in the community. So now, how do we classify parasites? Because as we go on in the lectures, you're going to find out that we have different types of parasites, and they have predilections for different tissues. The classification I'm giving you is from the medical perspective. So if you pick up a textbook of science, the classification is going to be different, okay? So this is a working diagnosis classification for medical parasitology. Is that clear? We can classify them broadly based on the number of cells that make up the parasite. When you have just one cell, we refer to them as protozoa. So protozoa are unicellular organisms. The entire organism is just one cell. But we can also have parasites that have many cells making up the entire organism. So when you have many cells, we say they are metazoa. M for many, M for metazoa, so many cells. And the metazoa are also referred to as the helminths, that is the worms. These are the worms. Now back to the unicellular organisms, because that's where we're going to start from. They are further subdivided into 4 groups based on the presence or absence of an organ of locomotion. So we have those that we refer to as sarcodina, or the amoebas. These are the organisms that move by producing false feet, what we call pseudopods. So they don't have a defined organ of locomotion as such, but the entire organism can flow and produce what we call pseudopodia. These are the amoeba. Then we have the sporozoa. These include organisms like Plasmodium parasite. They don't have a real defined organ of locomotion, but during some stage of development, they produce a lot of hydrolytic enzymes in certain organelles, which allows them to invade the host cell. So they are regarded as apicomplexas or sporozoas. Then we have the mastigophoras. These are organisms that produce flagella, and they move with the aid of that flagella that they produce. Then we have the ciliata or the ciliates. These are organisms that move using tiny ear-like structures that are referred to as cilia. They are not as long as flagella, but you have many of them, and the organism can use this to move around. So based on the organ of locomotion, we have 4 subdivisions of the unicellular parasites, Protozoa. Now, how can parasites access the human host? There are different routes because they are different parasites. So they use different strategies to invade the human host. Some can be inhaled. Organisms such as Acanthamoeba, Enterobius, and Naegleria fowleri can access the human body through the nose. Some of them are ingested in contaminated food or water, and you have quite a long list. We are going to discuss some of them. Some of them can be acquired through sexual contact, particularly Entamoeba species, Giardia species, and Trichomonas. Now, regarding Entamoeba and Giardia, let me quickly add that these are classically fecal-orally acquired. But because we now have people with different sexual orientation, men who have sex with men, people who practice oral sex, they are now regarded as being sexually acquired. So organisms such as Entamoeba and Giardia, which we're going to discuss in a few minutes, may be acquired through sexual contact, and they can also be acquired through the fecal-oral route. Some parasites are actually able to invade the intact skin. They can actually cut through the skin to invade the human body. These include examples like hookworm and schistosoma species. Then we have those that are vector-borne. So they are present in an insect vector. The insect comes around to have a blood meal and deposits the parasite. So they are acquired through inoculation from a vector. Then we can have contact and penetration of the eyes. And a very good example is the Acanthamoeba species. So these are different routes through which different parasites may be acquired. So this table is just to give you an idea of the predilection of different protozoan parasites for different tissues in the human body. Some parasites tend to be found in the gastrointestinal tract. Some are found in other body tissues such as the central nervous system, the eyes, or the genital tract. These include the group here. Acanthamoeba and Naegleria fowleri can invade the eyes. Naegleria fowleri can invade the central nervous system. Trichomonas vaginalis can invade the genital tract. We can also have parasites found in the blood tissue. These include things like Plasmodium, Trypanosoma, and Leishmania species. So different parasites, different sites of infection. So let's now focus on 2 parasites that are associated with the gastrointestinal tract. We start with Giardia lamblia. Now, this is a protozoan parasite. This is the adult form of the parasite. We refer to it as the trophozoite. Okay? Then we have what we call the cyst. So these are the two forms of the parasite. Remember I told you that they go through stages of development. So the matured form is the trophozoite. The cyst is the infective form. The trophozoite is the form that actively feeds and causes disease in humans. In terms of transmission, life cycle, and pathogenesis, the cysts are the infective form of the parasite. An infected human being will pass out both the trophozoite and the cyst in the stools. The trophozoites will die out within 30 minutes to 1 hour of being passed out into the environment because it cannot withstand the environmental stressors. However, the cyst has a thick covering, so it can resist environmental degradation and can persist in the environment. So if we have people defecating in the environment indiscriminately, these cysts are being deposited in the environment, and they are very tiny structures. And they can find their way into the food chain or into the water that we drink if we are not careful. And all it takes to have an infection is to drink the infective cyst in contaminated water or take it along in vegetables that are not properly washed and are eaten raw, and then infection is established. So when the cysts are ingested, excistation, that is the emergence of the trophozoite from the cyst, takes place in the duodenum. Now, unlike entamoeba histolytica, I want you to know that the trophozoites of Giardia lamblia does not invade the intestinal wall. As a result, a patient who has Giardia infection does not pass blood in the stool. The stools are non-bloody, okay? In terms of what the parasite does, when they multiply, they cause inflammation and they interfere with absorption of nutrients. So here you have the epidemiology of the parasite. The parasite is found worldwide. As long as you have people who are asymptomatic, people who defecate in the environment, the cyst is always present in the environment. Unfortunately, some people are infected, but they are not symptomatic. These are the people we worry about because they are the reservoir. Remember, we talked about the reservoir host. So they keep the parasite viable. They keep it in a form that it can infect another person, but they are not showing symptoms. So such a person can use the bathroom, did not wash his or her hands, and is offering you food. So the cyst, which you cannot see with your unaided eyes, is being offered to you in whatever was offered to you. With people who have immunoglobulin A deficiency, they are at a higher risk of coming down with infection. Remember that immunoglobulin A protects the mucosal surfaces. So deficiency of this immunoglobulin promotes infection. Outbreaks is related to contaminated water supplies. And one thing you should note is that chlorination does not kill the cyst. So the most important thing for the municipal water supply is not to have people contaminate the water source, because chlorination will not destroy it. It will not kill the cyst. So you can still turn on the taps and the cyst can still be delivered to you. But filtration can remove the cyst. So this is physical removal. Infection tends to be common in hikers who drink untreated stream water. because we can have animal reservoir for these parasites, and animals don't use the toilet. So they can continue to release the infective cyst in the environment, and this gets run off into the water. Now, many species of mammals are known as reservoirs apart from humans. Giardiasis is also common in male homosexuals because of their sexual orientation. The incidence is high among children in daycare, and this is because all it takes is for you to have an infected person in the daycare. This person will handle toys that children will put in their mouth, right? And the infected person can take care of a lot of kids, preparing their food and what have you. So it tends to be very common in children in daycare center where the hygiene is poor, and patients in mental health institutions because they can't take care of themselves properly. So how do you make a diagnosis? Because this is POD, we are not discussing the disease itself, okay? In other blocks, we are going to discuss this at higher levels, okay? So how do you make a diagnosis? Without looking at this slide, I'm sure most of you have. What clinical sample are you going to send to the lab? What clinical sample should be sent to the lab? Stool. Why? Because not only that, but why? Because yes, you can, but why do you think the cysts are present in the stool? Very good. The organism resides in the GI. So it makes sense. The infective form, the diagnostic form comes out in the stool. But the problem is you don't know how, when the cyst or the trophozoites are going to be passed in the stool. As a result, you have to evaluate at least 3 stool samples on different days so that you are sure that the patient is not infected, okay? So 1 stool sample is not enough. Now, what do you look out for? You look out for the cyst and the trophozoite if the stool sample gets to the lab on time. Within 30 minutes to 1 hour, the trophozoite will die out if it is not evaluated in the laboratory. So in the lab, if we get a fresh stool sample, we can actually see the motile trophozoites, which are pear-shaped, as you can see. They are pear-shaped, and they have flagella. Or we can look out for the cyst, which is characteristic of the organism. If you go back to this slide, yeah, it looks like the face of an old man. So we look out for this, okay? Another test you can perform is duodenal aspirate. And what you're trying to do is to pull up whatever is there in the duodenum. If the parasite is present, you can actually see the trophozoites in the duodenal aspirate. You can also perform the string test. It's no longer popular, but it can still be done, in which you give the patient a capsule to swallow, and after some hours, you pull out the capsule and you examine the content under the microscope. You can perform a biopsy of the upper small intestine, or you can perform fecal antigen tests using different approaches. You can also perform molecular assays such as DNA probes and polymerase chain reactions. The next organism we are going to discuss, which is also a GI protozoan parasite, is Entamoeba histolytica. And just like Giardia lamblia, there are just 2 stages to the development of the parasite. We have a cyst and a trophozoite, okay? But the cyst and trophozoites are different from that of Giardia lamblia, and that's why we can differentiate between the 2. The trophozoite is the motile form, just like what you have with Giardia, but Entamoeba is not a flagellate. It moves by production of pseudopodia, like I said earlier, false feet. The trophozoites can be found within the intestinal wall, or they can cause extraintestinal lesions. So unlike Giardia lamblia, Entamoeba histolytica can cause an invasive disease. So when the patient presents, they may tell you that initially the stool was non-bloody, but later it became bloody. So that's the cardinal difference between Giardia lamblia and Entamoeba histolytica diarrhea. Now the cyst can die in boiled water. It can be removed by filtration, but just like the cyst of Giardia lamblia, they are resistant to chlorination. So again, the municipal water supply must be protected from fecal contamination. But unlike Giardia lamblia, there are no animal reservoirs for Entamoeba histolytica. You only have humans as the host. Now, there are some characteristic features of the cyst that we look out for in the laboratory. When we look at the cyst, this is an example of a mature cyst. It tends to have what we call nuclei or karyosomes. There are 4 of them, okay? Then it has a rod-shaped chromatoidal body. This is characteristic of Entamoeba histolytica. When the cysts are ingested, they eventually give rise to the trophozoites. Although the excistation occurs in the small intestine, Entamoeba species tends to go and localize in the large intestine. In the large intestine, they can proliferate and invade, and at times, they can get into the blood circulation to cause extraintestinal infections, usually in the liver, the lungs, and occasionally in the central nervous system. So unlike Giardia lamblia, Entamoeba histolytica can cause both GI infection and extraintestinal infection, which can include organs such as the liver, the lungs, and the brain. Transmission is through fecal-oral route, when we have food or water contaminated with the infective cyst. And we are interested in the mature cyst. The cysts become infective when they become mature. That's when you have 4 nuclei present in the cyst. Now this is the typical flask-shaped ulcer of Entamoeba histolytica. It's called flask-shaped because you have a narrow invasion here, and then you have extensive destruction of the subepithelial tissue. So it's looking like a conical flask that you use in the chemistry lab. So that's why it's called the flask-shaped ulcer. Now, in terms of epidemiology, infection occurs worldwide, and it's frequently associated with tropical countries where you have poor sanitation and the environment is conducive. The cyst needs a warm and humid environment for it to persist for a very long time. Infection is also common in men who have sex with men. So how can you make a diagnosis? Again, because the organism is located in the GI, the stool sample is the best sample to send to the lab. And what do we look out for? We look out for the trophozoite and the cyst. We're going to focus on the features we are interested in a moment. There are other approaches to diagnosis. You can also perform a fecal antigen test like you have here, or you can perform molecular assays using polymerase chain reaction or a DNA probe. Now, when we say we look out for the trophozoite and the cyst, what are the features that helps us to distinguish the parasitic Entamoeba histolytica from other amoebic species that may be in the GI? Because you can have other Entamoeba species such as Entamoeba coli, which is a commensal in the gut. And unfortunately, it looks so much like Entamoeba histolytica unless you pay attention to certain features. So what are these features? The first one is the nature of the nucleus of the trophozoite. So if we get the trophozoite in the stool, so the stool was submitted to the lab within 30 minutes of collection, we can still see motile trophozoites. The first thing we look out for is the nature of the nucleus. Here the diagram is depicting what you have here. You can see that the chromatin granules are finely dispersed along the margins of the nuclear membrane. This is characteristic of the pathogenic strain. Whereas the non-pathogenic strain like you have here, they are not finely dispersed. You have them like clumps at the margins of the nuclear membrane. So that is something we look out for. Another thing is the size of the cyst and the number of its nuclei. So we can actually measure the size under the microscope, and we look out for the nuclei present in the cyst. As you can see here, this is a mature cyst of Entamoeba histolytica with 4 nuclei, whereas the non-pathogenic strain, as you can see, has more than 4. So these are features that we look out for to differentiate between the two. Another thing is the motility of the trophozoites. Remember that the trophozoites can move by producing pseudopodium. With the pathogenic strain, it is unidirectional. If you actually mount the trophozoite on the slide and the trophozoite is moving from left to right, it can actually move up the slide because it's not going to change direction. Whereas the non-pathogenic trophozoite is all over the place. It doesn't have a unidirectional motion. So these are things we look out for in the laboratory to differentiate the parasitic Entamoeba histolytica from the non-pathogenic strains that you could have in the stool sample. How can you prevent infection? Remember how it's transmitted: contamination of food and water. So using the bathroom, you must wash your hands. Because remember we said humans could be a reservoir of infection. So you could be infected, you're not showing symptoms, but you're passing out the infective form, which are the cysts. So washing your hands with soap and water is very important. Another thing is good food hygiene, particularly vegetables that we are going to consume raw. We are not going to cook them. So wash them with potable water, water that you are sure is not contaminated as well. Boil water, because we know chlorination does not kill the cyst. So if you are not too sure of your water source, your drinking water source, boil your water before drinking. We know that the infective form is passed out in the cyst. If we can break the chain of transmission by making sure that the environment is not fecally contaminated, we can prevent transmission of infection in a community. So provision of sanitary rooms so that people do not defecate in the environment. And of course, when you cook your food and you're not ready to eat immediately, make sure that your food is covered. Remember we said we can have mechanical hosts that will, transport hosts that can perch on feces in the environment and find their way into your kitchen. So whatever they picked up from the feces, they're going to deposit it on your cooked food that is not covered. So good food hygiene is very important in preventing infection. Any questions? Any questions? Okay.
Yes.
Okay. So have a good day.
Excuse me.
Yes.
What's the difference between parasitic host and accidental host and accidental parasite?
Accidental host, accidental parasite. Okay. Accidental host is not the usual host for the parasite. So for example, the dog tapeworm, it needs the dog to mature. As a larval form, we've not discussed development of worms, but let me just mention it here that unlike protozoan parasites, when worms develop, they develop from the egg through the larval stage. There are 4 larval stages. then they become the adult one. That's their usual life cycle. So if you have an accidental host exposed to one of the larval forms, it will not develop to the adult one. And because it can move around in the body and it has different antigens that the immune system, the host immune system can detect, it sets up an infective process in a way, okay? So because the host in which that parasite is found is not its usual host, it cannot develop further, okay? So that's the accidental host. Accidental parasite means the parasite is in the wrong host, okay? So the parasite is not dead, but the parasite cannot develop further. Has that helped you? If you don't understand very well now, don't worry. When we discuss helminths, I'm going to go over this again, okay? Yes.
So do parasites have structures for adherence?
For?
Adherence.
Yes, yes. They look at, there are certain receptors and there are certain nutrients that they require for their own development. Yes. Any other questions? Yes.
Doctor, why does the parasite live as two forms, cyst and adult?
Why do humans start as 2-egg stage, 4-egg stage?
So is the cyst just more mature than the adult?
No. The trophozoite, the actively feeding form, the form that replicates is the adult form, adult in quote. It's the most advanced form. The cyst is just a simple survival strategy phase of the parasite, okay? Because the parasite depends on the host for nutrients, which it cannot get in the environment. But if it exists as trophozoite in the environment, it's going to die off. The trophozoite is susceptible to temperature and lack of humidity, okay? But the cyst has a resistant covering, so it can withstand that, but it's not replicating. So it is regarded as the less matured form, okay? Although it is the less matured form, it is the infective form. The infective form means that that stage of the parasite can establish infection in a susceptible host, okay? The trophozoite cannot establish infection, but the trophozoite can replicate and produce the infective form. Does that make sense? Okay, so that's the cycle of development. Any other question? So have a good day. Okay, there's a question online. Can the human be a reservoir and a definitive host? Yes. We are the definitive host for the tapeworm, okay? We can be reservoir host for Giardia, lamblia, and Entamoeba histolytica. Being definitive host only means that you have the adult form of the parasite existing in that host, or you have sexual reproduction taking place in that host. Being a reservoir host means the parasite exists in a subclinical fashion, so the patient is not seeking treatment, but the parasite can still develop and the infective form is being shared from the reservoir host. It's like if you live in an environment where you don't have potable water and you have to store water. So you have a reservoir for the water, you take it when you need it. So that's what the reservoir host is doing essentially, keeping the parasite viable such that he or she can transmit it to the next susceptible host. Is that clear? Do I have any other questions? Sorry, I just want to be sure. So they are not the definitive host. Sama, I don't know what you mean. If you can explain further. Yeah, since it's in the GI, it will end in the stool, yes. Can the human, yeah, I think we've explained this. Can the human be a reservoir and a definitive host? Yes. Okay. So Sama, I don't understand your question. You can send me an email, okay? So I don't take too much time. So thank you, everybody.
Yeah, they're really cute, but then yeah, I think I'm good. Thank you though.
Don't end it.